This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 398. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi everybody, I'm Vincent Racaniello, and you are listening to the podcast all about viruses. A major new feature of the 4th edition of the textbook Principles of Virology is the addition of 26 video interviews with leading scientists who have made significant contributions to the field of virology. The idea is that for each chapter we give you a little bit of insight onto how some of the research was done. The interview you're going to hear today was done as part of that series, and it's included in the chapter on replication of DNA genomes. My guest is Sandra Weller, a professor in the Department of Molecular Biology and Biophysics at the University of Connecticut. Dr. Weller's laboratory uses genetic, biochemical, biophysical, structural, and molecular approaches to study mechanisms of synthesis, maturation, and cleavage and packaging of viral DNA genomes. Her research has led to the identification of a novel viral recombinase, the realization that host cell recombination and repair proteins are involved in herpes simplex virus DNA synthesis, also the discovery that host chaperones are rearranged in infected cells into foci, which act as quality control mechanisms for the production of properly folded proteins, and the finding that the initiation helicase origin binding protein UL9 is degraded during infection in a ubiquitin-mediated pathway. Dr. Weller, as you see, has made a lot of contributions to our understanding of how viral DNA genomes replicate. Let's go now to my conversation with Dr. Weller, which was recorded in Fort Collins, Colorado. I'm speaking with Professor Sandra Weller of the University of Connecticut. Thanks for joining me today, Sandy.
Thanks, Vince.
Can I call you Sandy?
You may.
This chat is about DNA replication, or I should say, this chat is for the chapter on DNA replication. I want to start out by asking you to tell us about where you were born and educated.
All right. Well, I was born in Pasadena, California.
That brings back all kinds of images.
Right. Well, I was a kind of surfer girl in the 60s. Then I went to Stanford, got my bachelor's.
We skipped high school. You went to high school in Pasadena?
Went to Blair High School, Pasadena, California.
Okay. What did you major in at Stanford?
I majored in biology and chemistry.
Okay.
And I got interested in viruses when I was in high school, actually.
How so?
I was dating a young man whose father was a virologist, and he was a retrovirologist. His name was Robert McAllister. He was hunting for human retroviruses. There weren't any at the time, but it was a very interesting project. And he had 6 children, none of whom were interested in science. And I was very interested in science. So he adopted me. I got to work in his lab and got very interested in retroviruses when I was in high school.
Wow.
But then I went to Stanford and I had to work on other things like bacterial membranes because I couldn't find any virologists. But I was still very interested in retroviruses. So when I was going to go to graduate school, I applied to University of Wisconsin, Madison, because I wanted to work for Howard Temin, who was working on retroviruses, avian retroviruses.
So you knew of Howard Temin from this?
I knew it from Dr. McAllister, yeah. And I either wanted to work for Howard Temin at Wisconsin or David Baltimore at MIT, but I ended up going to Wisconsin and studied retroviruses. As I was finishing that degree, I realized I wanted to... We still hadn't found any human retroviruses. Mm-hmm. This is during the 70s. There were no human retroviruses. I was studying an avian retrovirus, and there was a lot of work done in murine retroviruses, but there were no human retroviruses. And so I decided to change fields to do a postdoc because I wanted to work on a virus that caused human disease. Since retroviruses didn't cause human diseases, I had to leave retroviruses. I went to work in herpes, and I worked with Priscilla Schaefer at Harvard Medical School. 6 months after being a postdoc, after I started my postdoc, Bob Gallo came to the Dana-Farber and gave us a talk, and I realized, oh my goodness, there are human retroviruses. And it was sort of, I don't know if it was quite surprising because I had left the field because I wanted to study human pathogens, and I could have gone back into the retroviral field, but I also wanted to do genetics, and avian retroviruses had only 4 genes, and I wanted to do some more complicated genetics. So, and human retroviruses actually had a lot more. It would have been, but I had already moved. I moved on to herpes, and so I did a postdoc until about 1984. Moved to Connecticut, assistant professor, and
been there ever since.
Yeah.
I was going to ask you if you thought about going back at any point in your career. You could work on a retrovirus.
I could, but I sort of fell in love with herpes. Odd as that may sound.
But unlike love, herpes is forever.
Herpes is forever, right?
That's what Saul always says.
There are a number of jokes, most of which I would not tell on a tape. But
if Saul told them to, you couldn't repeat them. That's correct. Saul is a colleague of ours. Yeah. So it was this experience in high school working with a retrovirologist that got you interested in science?
No, I was interested in science in 7th grade. I had a particularly engaging teacher, Mr. McCoy, and he wanted us to do a science project. And being in Pasadena, I realized that Caltech was down the street, and I can't remember where I got the idea, but I wanted to work with fruit flies, so I was studying Drosophila. So I went over, and I ended up in Seymour Benzer's lab just to grab some. So I must have written ahead, and I picked up a vial of red-eyed fruit flies and a vial of white-eyed fruit flies, and I was going to do some genetics, and that led me on an incredible odyssey of learning how to cook because I had to make the media. I had to make the media that I could grow the flies in. So, my parents really thought I was crazy. My father was an attorney. My mother was a politico kind of person. So it was, having a daughter that was interested in science was totally outside their experience. They were very worried about me, actually, because they didn't really think I'd be able to find a career doing science. But, at a time when most mothers of teenage girls were tearing their hair because the girls are sullen, surly, and then wanting to be driven to the mall to go shopping, I asked to be driven to the scientific supply store to buy equipment to be able to make. I learned how to cook this way because I had to make the media, and it had to be just the right consistency. Because if it's too soft, the fruit flies would fly in it and they would die, and if it was too hard, the larvae couldn't do the crawling thing they have to do. So it was So I knew I wanted to do science when I was in the 8th grade.
So was there a point in your career where your parents were content that you had found something you liked?
No, even in high school, they were so nervous that they had both gone to UC Berkeley, and they called up a sorority sister of my mother's who had liked science, and she was a technician. Mm-hmm. And they said, "We're so worried. Sandy's really just seems to like science, and we don't think that there's a career there for a girl." I mean, they were really. And my father, he just was. He wanted there to be a career, so they were told that I could be a technician and there would be work in laboratories. So then they were okay. They were all right with it after that.
Was it exciting to work in Howard Temin's lab?
Absolutely. He was brilliant.
And it rubbed off on you?
Absolutely. Well, the one thing that did rub off on me, I think, is he was a very intuitive scientist, right? And his whole prediction that retroviruses would replicate through a DNA intermediate and that there would be an RNA-dependent DNA polymerase. A lot of that was very intuitive, and he was a very intuitive biologist. And I think sometimes scientists have to give themselves permission to be intuitive. Sometimes they get kind of locked into projects where they're going to do the next logical step and the next one and the next one, and I've found that the things that have given me the most satisfaction in my career are things where I kind of took a leap, a little bit of I wonder if that could be happening. So I think I like to think that I was able to sort of learn from Howard's. I don't think I will ever be as brilliant as he was, but I know I'm not. But and it was, we lost him way too early.
How did you get interested in DNA replication?
So when I went to Priscilla's lab as a postdoc, I initially wanted to work in herpes simplex virus because we thought they caused cervical cancer. In those days, it was right after a large epidemiological study had been done, and the correlations at the time were that in young girls, the age of first sexual experience and numbers of sexual partners predisposed cervical cancer. And at the time, we didn't know about papillomavirus. Right. And so the first idea was that it was herpes simplex, and people started working on fragments. It was right about the time when the genome was first cloned, and the fragments of the viral genome people were putting them into mouse 3T3 cells and looking for transformation. Yeah. And they actually thought that there were 2 regions of the herpes genome that they thought were transforming. And that was very exciting, so I thought I was going to work on cancer. One of the fragments that we thought might be transforming was one that contained 2 viral replication proteins, the DNA binding protein and the polymerase. And those 2 proteins are flanking or a region that we came to find was the origin. So I started working on that region of the genome and really just fell in love with sort of thinking about, we didn't know how many viral proteins were involved in DNA replication. We didn't know if the virus encoded the replication proteins or if there were cellular proteins. We knew that SV40 really only had one DNA replication protein that was virally encoded, and that it used the cellular proteins to replicate its genome. And we didn't know how many herpes viral proteins there were. And we had a lot of temperature-sensitive mutants. One of the reasons I went to Priscilla's lab was that she'd isolated dozens of temperature-sensitive mutants. They had a genetic map. They knew sort of the order of the genes, and we knew that the polymerase was next to the DNA binding protein, but we didn't have any physical mapping of the region, and we didn't really know we knew there could be an origin in that region, but we didn't know where it was. So I started doing physical mapping using marker rescue. And what I found was that the clones that I was using, all the restriction fragments that we cloned, contained deletions in this region. We could not make a full-length clone, which I turned out I figured out why, but it was sort of surprising. Why can't we clone this region? What's wrong? Why can't we propagate this region in bacteria? And I was doing marker rescue, trying to map the temperature-sensitive mutants, and I was able to map several temperature-sensitive mutants of the DNA binding protein, and I got very interested in why we couldn't clone that region. And that's how I met Max Gottesman. I was at a conference, and I reported that this was a very deletion-prone region, and I knew that there were several different deletions that could be made, some of them small, some of them large. And Max Gottesman heard me talk, and he said, I have a cloning strategy that I know is going to work. And so he said, come down to Washington. He was at the NIH at the time. He said, come down to Washington, and it was a lysogenic lambda cloning system where the idea was that if we could integrate this sequence into the lambda genome, or into the bacterial genome quickly enough, it maybe wouldn't delete. We figured that it was being deleted by E. coli recombination proteins, but the idea was that if you could make a lysogen, it wouldn't. So I spent several weeks cloning this thing, and it's still deleted. So then the next trial was to try to clone it in yeast. I thought, well, maybe yeast won't delete this sequence. So I collaborated with Andrew Murray, who was in Jack Shostak's lab at the time, and we did clone it in yeast, and we were able to make an undeleted clone. But by that time, I was just so curious about what was in this sequence that was causing deletions. So I decided I wanted to sequence it, and I couldn't get enough DNA out of the yeast to really sequence it. So I decided to sequence directly from the viral genome. And in those days, that was not easy to do, and we used Maxim and Gilbert sequencing. So I made lots of DNA and cut out the restriction fragments, labeled one end, and I went to Alan Maxim's lab and learned how to sequence. So collaborations obviously can be very important in one's career. So with a postdoc from Alan's lab, we were able to sequence that origin of replication. And I'll never forget one Saturday I was doing my reads. I was reading off the sequence. And I read a sequence, and then I read another region, and it was the same sequence. And I read another region, and it was the same sequence. And I realized that I had a totally perfect 144 base pair palindrome in this region. And no one had known that before. So 72 on a side. And not only that, but it contained many internal, indirect and direct repeats. So in the end, explaining why it was so deletion prone, because the coli enzymes were making deletions. And that was just a very exciting project to all of a sudden, So I cloned the first copy of that origin and realized why it was so deletion prone.
So you allowed your curiosity to lead you. Right. The curiosity about this origin and the flanking regions, and that's important in science, right?
Well, my career has taken me a lot of directions since I started that project, and when I went to start my own lab, I decided I want to keep working with DNA replication. And we only had 3 of the replication proteins at the time, and we figured there must be more. We had other DNA negative, temperature sensitive mutants that Priscilla had in her collection, and so I was able to map them all. And so I roughly figured out where the regions of the DNA replication proteins were. And I think we identified all 7 that way. And then Mark Chalberg came up with a transfection-based assay to map the actual 7 genes. And I remember conversations with him weekly. We'd call each other, and he'd say, I found another gene, and I said, I think I have a TS mutant in that. So we were able to really line up the clones that he was identifying using his transfection-based replication assay with the genetics telling us where the genes were. And as soon as we started, and then shortly thereafter, the sequence became available of the entire herpes genome. Duncan McGeoch from MRC Glasgow had sequenced the entire genome, and then we could line up our genes. Right. And that one looks like it might be a helicase. That one looks like it might be, a DNA binding protein. And ever since, I've sort of let the virus lead me into, we started doing replication biochemistry, because once we saw that we might have a helicase, then we had to express that protein in E. coli, and eventually had to express it in baculo and insect cells in order to make a functional protein. Then we realized that it was making a complex of 3 proteins. The helicase primase had 3 subunits. And then we started doing cell biology. So really, every time, we had a question, we had to learn a brand new technology in order to do it. So I was very lucky to be in the department I was in, because we had a lot of really good biochemists. I'd been a geneticist up to that point. I was interested in mapping genes, and once we'd mapped the ones we were interested in, I realized, now I have to become a biochemist. And luckily, I was in a really good department that I could do that, and purified proteins and started doing helicase assays, and I still am very interested in those proteins. So,
can you point out a key experiment from this time having to do with DNA replication?
Well, one thing that we've become very curious about, and even we're at the beginning, has to do with the mechanism of replication. So the replication systems that had been studied up until the point of the 80s when I started my own lab were SV40, which is a circular DNA molecule, which has an origin of replication. The antigen binds to it, replicates bidirectionally all around the circle. You get 2 concatenamers at the end. It's very orderly. They separate. Now you have 2 daughter strands. And that seemed like it's a very simple, elegant model. Herpes is not that well behaved. So it's a linear genome, and although there has been a model that proposed that the viral genome circularizes and that rolling circle replication occurs like lambda phage, we really don't have any direct evidence that the viral genome circularizes, and in fact, we don't think rolling circle replication is the primary mode of replication. I can't say that it never happens, but I don't think it's the primary mode. And we think recombination plays a big role in this. And we have now been able to isolate a viral recombinase that we think is analogous, and we know it is analogous to the lambda system, actually. Lambda, the lambda red alpha beta proteins, which can do recombinating, herpes has an exo that's related to lambda exo, and it has a DNA binding protein that actually acts as a single-strand annealing protein. So it has an exo plus a single-strand annealing protein, and that's really all you need to do single-strand annealing. So we actually think now that the virus uses single-strand annealing to replicate its genome, and it's not necessarily rolling circle. And although it's still controversial, and we haven't nailed all the nails into the coffin in terms of proving it, we believe that it's using a really fundamentally different mechanism of DNA replication from all other viruses, except maybe lambda may also use this kind of mechanism.
Are you still continuing to work on this problem today?
Absolutely. It's one of the most exciting ones that we do.
So can you give us some idea of some of the things you're doing?
Well, this recombinase, which is made up of a nuclease and a single-strand binding protein, that single-strand binding protein is the same single-strand binding protein that is used for replication. So it's actually used in both processes. And we're very interested. That protein by itself can form filaments in solution, and it can also form a filament on a single-strand DNA. And we're very interested in how that ability to form filaments relates to its other properties in the cell. It's a multifunctional protein. It's involved in DNA replication. It's involved in recombination. It's also involved in how the virus manipulates the nucleus. This virus totally reorganizes the nucleus. So it makes these very large replication compartments that take up almost the entire volume of the nucleus. And one question we have right now is what's the nature of those replication compartments and how are they forming? And we think that the ability of ICP8 to form filaments may be related to its ability to, these compartments take up almost the entire nucleus, even when DNA replication isn't happening. So how would that kind of space filling occur? And what we think is happening is that we have a series of filaments that are forming kind of a meshwork of ICP8, maybe along with multivalent weak interactions that are forming kind of a scaffold on which other replication proteins can form, and then eventually replication forks can form. So we're very interested in replication fork assembly, how it forms in the cell, maybe on some of these matrices, if the filaments are forming matrices. We're also interested in these replication compartments. You know, you've seen IF pictures of them, I'm sure, and I think there was one from my lab in the book the last time. And these compartments look like they might have membranes around them. They just look so delineated from the rest of the nucleus. And you think, well, we know there's no membrane, but how would a structure form that has such a tight boundary between where there is ICP8 and where there isn't? And so we think we're very excited about sort of the biophysical properties that would allow this to happen. And even some of the work that we heard at this meeting about phase transitions and things like stress granules that are made up of complexes of proteins and in some cases nucleic acid that form these loose networks that actually allow you to form something that looks like an organelle. And you know, cells are not just bags of enzymes. There's so much structure to be learned about. So we're very interested in what kind of structures are being made and if they really need filaments. And now we have mutants that don't make filaments, and we're trying to figure out what's needed in ICP8 to make a filament. And we'd really like to do some in terms of technology that's new that we can use. I think we'd like to use some much higher resolution microscopy that might actually help us look in more detail at what kind of filaments are being formed and how that affects the in vivo in an infected cell, how that affects the replication compartment formation. You
should talk to Claude O'Shea.
I just spent 3 hours with Claude O'Shea just now.
Because filaments are...
No, in fact, that cell paper that she wrote is one that we read all over and over and over again. And so we were just showing her... Actually, in fact, my student gave a talk at this meeting, and Claude approached us afterwards, and I've known Claude for years, so we had a great conversation.
This episode is sponsored by CuriosityStream, the world's first ad-free nonfiction streaming service with over 1,400 titles and 600 hours of content. It was founded by John Hendricks of Discovery Communications, so you're sure to be getting real science shows, not reality TV shows, currently ruining cable TV. You can get these streaming movies on many platforms, the web, Roku, Android, iOS, Chromecast, Amazon Fire, Amazon Kindle, Apple TV, and you can get it in 196 countries worldwide. And what do they have? A wide variety of science and technology content. In addition, they have nature, history, and much more, all nonfiction. The library of interviews and lectures includes Stephen Hawking's Universe, a series where Hawking traces the history of astronomical theories and technology. A movie called Viruses, Destruction and Creation. This is a Curiosity Stream original in which doctors, virologists, and scientists examine the spread of Zika virus, what effect this epidemic may have on large populations, and possible next steps to stop the illness. Life on Us, a series that explores the biodiversity of our bodies. Secret Life Underground, discover the secret world of organisms that populate the underground world. Monthly and annual plans are available, starting at just $2.99 a month, less than a cup of coffee. Enrich your life and continue learning with one of the largest 4K libraries on the net, over 50-plus hours of 4K content. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV.
How has technology changed in the years that you've been working in ways that you have used?
So, at every step, you tend of your career, you tend to be limited by not having something. So, right before I joined Priscilla's lab, and they had all these temperature-sensitive mutants, and they mapped them by recombination. So, you infect a cell with 2 temperature-sensitive mutants and measure the frequency of recombination, and then recombinant DNA came out. So then we had clones of each of the restriction fragments from across the genome, and we could do actual marker rescue by mixing the TS mutant DNA with the small fragments. So that was high technology in the mid 80s, and around that time, people started doing confocal microscopy and using immunofluorescence, and so we were able to see replication compartments because we had antibodies to the different DNA. David Knipe did that initial work, and we were able to do that. So even just the crudest confocal microscopes told us a lot. But I'm seeing a lot of from my own work, the value of some of microscopy is just exploding in terms of its ability to see smaller and smaller, with higher and higher resolution, to see structures that almost at the atomic level. I mean, it's really amazing that people are, I don't want to say this in a way that my crystallographer colleagues will hear about, but microscopy could replace crystallography in a lot of ways. So certainly the microscopy, the ability to do proteomics and look at established networks of protein interactions, because I didn't mention it, but we're also very interested in the host cell responses to these viruses and looking at which cellular proteins are coming into the viral replication compartments, and then being able to confirm direct protein-protein interactions using a number of methods, and we're doing some biophysical analysis. We're looking at oligomerization using different new kinds of dynamic light scattering. There's really beautiful instruments for making it a lot easier. We still do analytical ultracentrifugation, which is a rather old school technique, but we also can now do fluorescence correlation spectroscopy and dynamic light scattering, and it's really been enjoyable for me to learn each time, to realize that there's something new out there that we can apply. And who knows what's going to be in the next 5 or 10 years that we couldn't even think about now. But it really does change the way you hit a roadblock, and you can't solve that problem until a new technology comes out, and then.
You don't use a Model E, do you? Analytical centrifugation.
I did when I was a graduate student. I actually send my sample over to my colleague over on the Storrs campus who runs the National Labs for Analytical Ultracentrifugation, and I haven't actually seen, I don't think it's a Model E.
Probably not, yeah.
Probably not.
But you remember them, right?
I absolutely remember
Elephants,
them.
huge.
Yeah, no, I took up the entire room.
Yeah, it's another trend, miniaturization, right?
Yes, yeah.
It's amazing.
No, it's true.
So of all the work you've done in science, of all sorts, what would you single out as that which has had the most effect on the field?
Well, I'm not sure if my work had an effect on this field, but I'll tell you the project that thrilled me the most when it was finally, and it has to do with new technologies telling you something that you hit a roadblock. And so when I was a graduate student, I studied avian retroviruses, and I discovered that some of them were cytopathic. They could kill cells. And that wasn't known. Retroviruses are not normally thought of as being cytopathic. They can go in and they can integrate, and they do so in a way that usually doesn't kill the cell. So it was kind of an unusual finding. In fact, when I first made that observation, I remember Howard being very skeptical, Dr. Tubman being very skeptical of it. And, I don't think he ever said this, but I definitely got the feeling that he thought that retroviruses don't kill cells. First-year graduate students do kill cells. But I was right. The virus was killing the cell. I finally was able to convince him. And I mapped there were different subgroups. There was a genetic system of avian leukosis viruses. Some of them could use one receptor, some could use another receptor. And there were some subgroups were cytopathic and some of them weren't. And I discovered that the cytopathic retroviruses had a very unusual property of when I separated the cells that were dead from the cells that were alive, the dead cells had this ladder of DNA if I ran a DNA gel. And so the cellular DNA was becoming fragmented. And it really was a surprising finding, and we didn't know what it was, and Howard didn't want to publish it, but I said, I think we should publish this because someday somebody will figure this out. Someday somebody will figure out why this happened. And sure enough, 16 years later, I was organizing a FACID meeting and invited John Young to speak, and I'd never met John. I knew that he and Paul Bates had cloned the first avian leukosis virus receptors, and the first one they cloned was a non-cytopathic receptor. And I was hoping that they would clone a cytopathic receptor, and they hadn't yet. They did this when they were in Harold Varmus's lab. But he stood up and he said, 16 years ago, Sandy Weller published the first apoptotic ladder and the first fragmented ladder. And he said, now we have cloned the receptor for that virus, and it's a TNF family member and induces apoptosis. So I believe that I discovered apoptosis, but I had no clue what it was, and I had no way of knowing what it was, and it wasn't until John was able to clone the receptor and the technology had advanced so that they could, do a whole cDNA clone of cells and figure out what was the actual receptor that we realized what it was.
Of course, at the time, no one knew about apoptosis at all, right?
No, when I was doing our work, but it made a perfect story that the very receptor that was used, and he proved that in a ligand-specific way you could induce apoptosis by just treating it with the glycoprotein from that particular retrovirus.
Right. That's great.
And the saddest thing about that was that Howard had actually passed away 2 years before that, and it would have been a story that he would have absolutely loved.
At least he let you publish it, right?
He did let me publish it, and so
that's good.
So I just was on cloud nine that finally that had been solved.
That's a good story. So if you had not become a scientist, what else would you have done?
At the time, I thought I would become a lawyer, and I think that was because my father was a lawyer, and my father, I liked math, and I was good at math, and my father convinced me, and I think it's true that a lot of attorneys are very logical, and he had always loved math as well. So I sort of thought, well, that would be something that I could I could do with that interest of mine. At the time, actually, I should say that he was an attorney and he had accountants that worked in his office. And so he actually wanted me to become an accountant because I could use the math. But that didn't thrill me so much. But in recent years, I've just decided that if I was going to do something else, I would probably want to be a psychologist. Because it's just, after you've been in this field for a while and you've had a lot of students and you've had a lot of colleagues, I am also a chair of my department, and the more you deal with people, the more you're dealing with different kinds of people, and just trying to figure out what makes different people tick, what makes some students motivated by certain things, how to motivate each student, how to some students love it when you look over their shoulder every time they're developing a new gel and try to discuss with them right away what their I hated that. I hated it for myself. If I took a film off the exomat and was trying to figure out what it was, if someone asked me what the result was at that point, I was furious. Give me time. So some people like to be kind of coddled and paid a lot of attention to. Others just want to be independent and don't want to tell you their results until they already figure it out. So just looking at different, people. So
figure out why that is, right?
Why do people? I don't know. I find people fascinating. So,
what advice would you have for readers of this book who are thinking about having a career in science?
That's a good question. First of all, I think the book's fabulous. I teach it. I use it in my teaching, and I really like the way that chapters are laid out and important observations are put in boxes so that the students can see them. And very often, if we have time, we go back and we read those papers. And I think it really makes a lot of sense to try to go back to that original literature and sort of say, it's sort of classic papers, and some of these papers are, to go back to when people were making their own isotopes and some of the really classic, Messelson-Stahl experiments. But it gives you an appreciation for how far we've come when you read some of these old papers, and you realize a lot of things. First of all, how persistent they had to be in order to, there was no such thing as going to the refrigerator and freezer and pulling out a restriction enzyme. I mean, if you, we used to purify them ourselves because we didn't have.
Yeah, Mike Bishop told us how he used to make S35 methionine.
Yeah, no, that's right. That's right. So the farther you go back, the more surprising it is of things that we take for granted of, we're just going to call it biolabs and they're going to deliver this and that and they'll bring it in the buffer that you need.
But it is good to have things made so you can spend time doing it.
Well, it certainly gives you more time to think about the experiments. And I know from even this last class I've taught with using the book is that it's really, I think students really do get excited about virology. Virology is still very important, and just learning about how viruses have evolved to take advantage of the cells that they're in and evade host cell immunity and establish what they establish. And herpes viruses have been with us since we were us. I mean, they've really, the coevolution between us and the viruses have resulted in this kind of evolutionary arms race that is very fascinating now to look at. And I think I would certainly recommend that students pursue a career in this field. It's a very rewarding one. There's always things to learn. We certainly have only scratched the surface, and it's important work. Viruses are still killing us. They're still killing a lot of people. Yeah. It's funny every time you think about an accomplishment we've made in this field. We almost eradicated polio. Maybe we have now, but.
That's still almost.
It's still almost, and smallpox, and then even in the late 70s, when I said, "Well, we don't have any human retroviruses." Well, starting in 1980, we have a really remarkably serious epidemic on our hands that we still haven't solved, and we still don't have a vaccine, and we have some pretty good drugs, but we still have a lot to learn. And not to mention the fact that viruses have taught us almost everything that we know about eukaryotic cells. I mean, we learned about splicing, we learned about intracellular transport, we learned about how receptors work, we learned about DNA replication. Even cellular DNA replication was discovered only because by SV40, because SV40 was utilizing the cellular factors. So hopefully students will just realize how wonderful viruses are.
How important is a good mentor for students?
Mentors are very important. Howard was a very different kind of mentor than... Howard was very formal, and one thing that happened when I was working with Howard is I was his first female student. Mm. And that made him very nervous. And at the time I was sort of surprised by it because I didn't really see... I'd been an undergraduate at Stanford. I never felt obvious effects of sexism or people that didn't think I was good enough. I mean, it seemed like I'd never really run across gender discrimination. And yet he, either because he was married to a geneticist, Rayla was a first-class geneticist herself, and maybe she was advising him, but when I first started in the lab, he was worried that he wouldn't be a good mentor to me, only because he wasn't female. And he insisted that my entire thesis committee be female, because he wanted to provide me with role models. And at the time I thought that was very silly, because I really hadn't seen the effects of gender discrimination. And by the time I left, I actually was very grateful for him for having done that. And it didn't work out that I kept the same. I mean, we had a lot of female virologists, like Martha Howe and Mary Osborne, and there were some good mentors, but it struck me as a very kind and a thoughtful thing for him to do, to realize that, and there certainly are lifestyle choices that you have to think about, and it is harder and there is a lot of gender bias still in our field, and so having role models of people that, part of the reason for the gender discrimination is that women do leave the pipeline because they want to have families and they think they can't have a family if they do this career. I have 2 children. It's been a wonderful thing for me to be able to do that. Not an easy thing, but I hope that by realizing that I've been able to do that and raise 2 wonderful boys, that it is possible to have a career. But it's not, there are sacrifices.
Sure.
There were days that I felt like I wasn't being a good mom if I was doing my experiments, and I wasn't being a good scientist if I was going over to a soccer game. So it's... But having role models, having people to talk to, having people to help you at different stages of your career. Now that I'm a chair, I try to be a good mentor to my faculty, and I read their grants. I try to promote them, and giving talks at national meetings, and just having somebody that can just be thoughtful about talking to you about career development is very important. Being a role model. Sometimes you learn what kind of a mentor, what kind of scientist to be by either watching your mentor and realizing you don't want to be like that person, or that you do. But after I graduated, Howard Temin was a very good mentor, but he was very formal when we were in his lab. So as a student, we called him Dr. Temin, and it was a very formal relationship. And I never forget the day after I defended my thesis, there's a note on my desk signed Howard. And after that, he was a friend. But he took that job extremely seriously, and he wanted to be a good mentor. But after I left, he would invite me back and give me seminars and gave me a lot of opportunities.
Yeah. So for a young person entering the field, students, postdocs even, how do you identify this good mentor? What are the important qualities?
Well, someone who's willing to talk to you and talk to you about career development would be, there are some people in this world, as we were talking about understanding people, who are more interested in themselves than they are in promoting someone else. So those people are kind of toxic, and hopefully young students would know enough, or would get a feeling of that person may not be interested in me as a person. I want to work with someone who's interested in my career development. A good teacher.
Sure.
And I don't think there's just one good mentor. I mean, even though you pick a thesis advisor who's going to be a mentor, you need to have multiple mentors. So, and I think students should choose their advisory committees based on being able to know that they can go into that person's office anytime and say, I'm having this problem with this gradient. Can you help me out? Or I need to decide whether I should go into, should I do a postdoc? Should I go into industry? Just people that you can talk to. I think mentoring is really critical. And I think it's important for us as mentors to really just be able to listen. Sometimes I don't listen as well as I should. I'm feeling like I should always be there just to be a person that they can talk to. And it's not always going to be your thesis. Your best mentor may not be your thesis advisor or your postdoctoral mentor, but another colleague,
Sure.
Peer mentors are important. The students that come in together, they're often very supportive for each other. But it's important to have mentors. And in these days, it's really important to have people that will help you out in a lot of ways, like reading your papers, reading your grants. This is not a time for cowboy science, or I'm doing it on my own, I don't need any help. It's just really not a time for that, because with the funding climate being as hard as it is, it's really important to craft a grant, and the way to do it, to craft the best grant, is to have people that are willing to look at it for you. And it's time consuming. It takes a lot of time.
I've been speaking with Professor Sandra Weller of the University of Connecticut. Thanks for talking with me today, Sandy.
Thanks, it was a lot of fun.
Hope you enjoyed my chat with Sandy Weller. You'll be able to find this episode of TWiV and all the others at iTunes and also at microbe.tv/TWiV. And of course, as always, we love getting your questions and comments. Please send them to TWiV at microbe.tv. If you'd like to support our work here at TWiV and also the other science shows of Microbe TV, become a patron. You can subscribe monthly for as little as $1. And to do that, you can go to patreon.com/microbetv. That's patreon.com/microbetv. Or if you want to contribute in other ways, check out microbe.tv/contribute, where we have PayPal and credit card methods available. I want to thank Sandy Weller for participating in this interview. As I said, it was recorded in Fort Collins, Colorado, during the annual meeting of the American Society for Virology. And I want to thank ASM Press, which is part of the American Society for Microbiology, and the director of ASM Press, Christine Charlop, for letting us do these interviews. We pitched the idea to her. She thought it was a good idea. She facilitated everything and included it in the book. So if you do buy the textbooks, you'll find links to these interviews, videos, as well as audio in each chapter. I also want to thank Ray Ortega, a media specialist with ASM, who went with me to Fort Collins to record several interviews, including this one with Sandy Weller, and also Chris Condayan of ASM for production. As I said, you can see a video of this episode through the links in the textbook. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.